Hello and welcome to your next Life Sciences lesson. Today we are going to be looking at the topic of plant anatomy. We're going to start off with a question to focus our minds. Why do the different parts of a plant look different if they are all made of plant cells? This is the concept map showing what we are covering under plant anatomy. We did start looking at plant anatomy. In our last lesson, we looked at roots, and today we are going to be doing the next two topics, stems and leaves. However, we need to start by revising the types of cells that we would find in a plant. So, let's revise the different types of plants or the different types of cells. In every plant, we have some cells which are meristematic. Remember, they keep dividing, and they are like tiny baby cells. Then, on the outside of each part of the plant, we have epidermal cells. Sometimes they have a cuticle, which is waxy to prevent water loss. Sometimes they have root hairs, and we talked about those in the last lesson because they are found on the roots to absorb water. Many epidermises also have openings in them called stomata. And those are little openings through which a leaf and a stem can absorb gases. Then, most of the inside of a plant is filled with something called ground tissue. And these are ordinary cells that carry out many different functions. The most common type is parenchyma, and that is parenchyma. Large cells with thin walls. They're great for storage, but not very good for support. If the plant needs support, it will have collenchyma cells, for example, those which have thickened corners for strength, or if it really needs lots of strength, they would have sclerenchyma cells, which have walls that are thickened all the way around. Then, the last two types of cells are your vascular tissue, which is xylem and phloem. Here is the xylem, and its function is to transport water. That is the phloem, and the phloem's function is to transport food that has already been manufactured around the plant. We also need to revise, what does a plant look like? What is it made of? And right in primary school, you looked at something like this. Plants have roots, stems, and leaves, and at certain times of the year, they will also have flowers. Now, every single one of these has got a specific function to carry out. In the last lesson, we looked at the root with its functions of anchoring water absorption and food storage. What we're going to look at today is the stem, and most stems grow upright, so they would need to support the plant. If we look at this plant is standing upright, and it also is going to need water coming from the soil to go up the plant. So, what types of tissues would we need to find in each of these parts of a plant? A stem If its function is support, what types of cell is it going to need? Parenchyma or sclerenchyma. If its function is transport, what is it going to need? Xylem and phloem. So what we're going to be looking at, we're going to be looking at what do we find in a stem. Not only what types of cells do we find, but where they are. In other words, how they are arranged. Before we start going into detail, let's take a break to make sure your brains are fresh for the next section. Welcome back from your break. I hope your brains are now fresh and ready to absorb more plant anatomy. We are going to start by looking at the structure of a stem. We've already revised the different types of tissues, so we're going to look at what types of tissues we find in a stem, what they do there, and how they are arranged. What tissues will be needed in a stem? Here is your stem. Let's think. Does it need meristematic tissue? Will it need to grow? Does it need epidermal tissue? And if so, which ones? Would we find parenchyma, collenchyma, sclerenchyma, and xylem, and phloem? And if so, what do these cells actually do? We would find meristematic cells in buds. That's a bud. And that is where new leaves and flowers grow. We would also find epidermal cells, and quite important, they would cover the outside of the stem, and they would need a cuticle to prevent water loss, plus they would need stomata to get in the gases they need. Would we need parenchyma? Most definitely, to fill spaces and store food. Then, our supporting tissues. Collenchyma. Are we going to need collenchyma? Very definitely. That is what keeps the stem standing upright. Some plants only have collenchyma for support. Others also have sclerenchyma. And remember, sclerenchyma is much, much tougher, so it does a better job of support. Then, because one of the functions of the stem is to take water upwards and food downwards, we would find xylem, which is our water transport tissue, and phloem, which is our food transport tissue. So these are the cells that we're going to find in a stem. The next question is, well, where in a stem will we actually find all these tissues? This is a cross section through a stem. In other words, we've taken a stem and we've cut it across. And if you look at it, it's very, very confusing to start. But let's start with the easy things, the things that are easy to recognize. And obviously, the first one that's easy to recognize is, does it have a skin? And the skin is the epidermis. Then there are a whole lot of big cells in the center, and if you look at them, they've got thin walls. What type of cells would those be? Those would be parenchyma cells. Large cells, thin walls, with air spaces between them. Then the rest all looks pretty confusing. How can we make sense of it? Remember, the darker the color, so if we look there, the color is quite dark. That shows that the cells have got thick walls. There, the color is also dark, so those are cells with thick walls. If we look here, these cells also have thick walls. And if we have a closer look, we're going to be able to identify which of the cells with thick walls they actually are. If we look at the next picture, this looks kind of different and sort of the same to the previous one. And the reason is that the two are sections of different stems. So every stem would look slightly different, but if you look, there is the same pattern. And that's what we're looking for, the pattern for the layout of these different tissues. Another thing, the color is different. Look at that color. And if we look at this picture, look at that color. And the reason why the colors are different is simply because these two pictures have been stained by different dyes. Because most of the cell walls and things like that in a plant are colorless, so we need to stain them so we can see them clearly. And the different colors actually simply are the different stains that have been used to bring up the different types of tissues. So what we're going to look at now is we've already found the epidermis. Sure, that was the easy one. And we have already found the parenchyma cells in the center, and that area of parenchyma cells is called the pith. Then we were a little bit confused by all the different cells with thick walls. Now, if we look at that pattern, you see there are a whole lot of those. And those are all vascular bundles. You can see all the way around near the outside, there are a whole lot of these vascular bundles, and that is where the xylem and phloem is found. Then, just underneath the epidermis, the cells there you can see are it's quite dark, meaning the cells have got thick walls, and that is your collenchyma, and that makes sense because the collenchyma is there for support. So we have identified collenchyma. So let's look at the next picture, and that puts in words the fact that there's collenchyma under the epidermis, there's vascular tissue, they're called vascular bundles, and those are the little patches of xylem and phloem, and then the center of the stem with the big parenchyma cells, that is called the pith. I know it's very confusing looking at all these cells everywhere. So what we're going to do now is we're going to look at a simpler diagram which simply shows us where the different types of tissues are found, and hopefully that will make it a bit easier for you to understand the pattern we find in the stem of a plant. This is a section across a plant, and that bit there is the cross section. You note there's no detail of the cells. It's simply showing us where the different types of cells are found. And something like that is one of the vascular bundles of xylem and phloem. Here, the section has been cut downwards. So here you are looking at the xylem vessels from the side. So this is called a longitudinal or lengthways section. Then, looking at the cell from the outside is called a side view. So this diagram shows us 3 different views of the arrangement of tissues in a stem. The part that is most confusing is these vascular bundle things. So what we're going to look at now is we're going to up the magnification of the microscope and look at only part of a stem, but it means we can see the vascular bundle in quite a bit of detail, which makes it much clearer and much easier to work out what is going on. This circled area is the vascular bundle. You can probably pick up, there's the epidermis, there's the collenchyma, but here we're focusing on the vascular bundle. We can see here nice big empty cells, and those would be your xylem cells. They've also got thick walls. Then what you see here are smaller cells which also have thick walls. And their walls are evenly thickened, so that is the sclerenchyma. And between the two are thin-walled cells. And there's 2 different types of thin-walled cells here, filling that space of thin-walled cells. That is the phloem. And you can pick up there are little companion cells between the big sieve tube cells. Then, below that is a layer of flattened elongated cells, which we haven't come across before. Those are meristematic cells, and they form what we call the cambium, and that is what enables a plant, as it gets bigger, to grow more xylem and phloem, because if it's bigger, it's going to need to transport more water upwards because it's going to have more leaves. This is simply another picture. And if we look at it, there are the fibers. Why do they look so different? A different dye has been used so they show up as a different color. There's the phloem with the big sieve tube cells and the small companion cells with thin walls. There's the vascular cambium, the flattened cells, and here we can find our nice big xylem cells. It's often easier to understand by drawing a plan or a line diagram of a stem. And a line diagram simply shows where the different types of cells are found, doesn't actually tell you what the cells look like. So, let's have a look at this diagram of a plan and see if we can recognize all the different regions. Often it's easier to look at a plan diagram which shows you where the different types of cells are found without actually giving you any detail of what the cells look like. This diagram is a simplified diagram showing vascular bundles inside the plant stem. Let's now have a look and see what can we identify from what we have already talked about. First one is these things here, okay? And these, at the tip of the vascular bundles, those are patches of xylem. What else do we find in our vascular bundles? We find phloem, and that's where the phloem is found. So that would be an area of phloem. Then we would also find the bundle cap of sclerenchyma, The last part of the vascular bundle is this area here between the xylem and the phloem, and that is where the cambium or meristematic tissue is found. So we've now looked at the detail of the vascular bundle. We're now going to look at some of the other parts that we can recognize. Easiest one to recognize always is the epidermis, because every plant part has to have an epidermis. Also, every plant part will have cells filling in spaces, and those would be parenchyma cells, and the parenchyma cells are found in the middle with the pith. This particular diagram doesn't show the collenchyma. Next picture we're going on to is a drawn diagram showing the detail of a vascular bundle, and let's see if we can pick up the different parts from this. Easiest one is always xylem. Very easy to pick up, large cells with extra thick walls. Next easiest one to pick up is going to be your phloem, and we can pick that up because there are some big cells, which are the sieve tube cells, surrounded by smaller cells, which are the companion cells. Another easy one to identify is this part here, which is the bundle cap, and that has got cells with thick walls, but they are small cells. You can see the difference between the size of a xylem cell and the size of one of those sclerenchyma cells. That's the way you can tell the difference, because they both have thick walls. Then we can also find the other parts of the stem, and this one shows very, very nicely. Here we have the epidermis, and this also shows very nicely the collenchyma with the thickened corners. It also shows very nicely a whole lot of parenchyma cells between the vascular bundles and filling the center. The parenchyma cells that are between the vascular bundles form part of the cortex, whereas those right in the center form part of the pith. There's really not much difference in their structure, it's just where they are found. Welcome back to our Life Sciences lesson on plant anatomy. I hope your brains are fresh and you're ready to learn interesting new things. Previously, we have covered parts of the plant. We've looked at what does a root look like if we cut it open. We've looked at what does a stem look like if you cut it open. And the focus of this segment, we're going to look at leaves. If you cut open a leaf, what do you actually see? What types of cells will be there and how will they be arranged? To set the scene, we need to revise the structure of a plant. Right from primary school, you know plants consist of roots, stems, and leaves. And earlier on, we looked at the functions of these different parts, and we're now going to be focusing on what does a leaf do. The three functions of a leaf: photosynthesis, gaseous exchange, and transpiration. And to be able to do those 3 functions, the leaf has to be specially structured so it can carry out all 3 of them. So let's have a look. To be able to carry out these functions, which tissues will a leaf need? Meristematic, epidermal. Obviously, epidermal, they all need epidermal, so that's easy. Does it need meristematic tissue? Do leaves keep growing bigger and bigger? No, they don't. Once a leaf is formed, it's formed. So it doesn't have meristematic tissue, but it definitely needs epidermal tissue. Then, will it need parenchyma? Yes, it will. Will it need something to support it, either collenchyma or sclerenchyma? Yes, it will. Will it need xylem and phloem? Most definitely, because xylem and phloem do the transporting. So, let's look at that. A leaf will have all of the types of tissue except the meristematic tissue. Epidermal, parenchyma, collenchyma. Most leaves don't have sclerenchyma because collenchyma does the job for them, and then of course xylem and phloem. If we look at a leaf, if you look at the top surface of a leaf and the bottom surface of the same leaf, you'll notice the colors are different. And that is because the upper part of the leaf gets lots and lots of sunlight, so it does lots and lots of photosynthesizing, but the bottom part of the leaf doesn't get direct sunlight, so it has less photosynthesizing to do, so it actually has less chloroplasts, which gives it that much, much lighter color. But all parts of the leaf will have chloroplasts in them. Next one. Stems and roots are all round. So why is the leaf flat? And the reason is the leaf's main function is photosynthesis. That's what keeps life on Earth going. The fact that plants make food for all the animals that live on Earth. And the more food the plants can make, the more they can grow. So if we look at a leaf, it's got a broad, flat leaf blade. And that leaf blade is flat and thin, and the reason is it has a large surface area so that when the sun's rays hit it, the leaf can absorb as much sunlight as possible so it can do as much photosynthesizing as possible. Another point is that being flat means that all the cells are close to the surface so that all the cells can get gas. Because for photosynthesis, from previous years, you should know that for photosynthesis, cells actually need carbon dioxide, and being flat means they can absorb the carbon dioxide easily. Next question is, why do leaves have so many veins? If we look at these pictures, those are called leaf skeletons, and what has happened is that the soft-walled cells in the leaf have died, and the only things that remain are those with extremely thick walls, in other words, collenchyma, sclerenchyma, and xylem. And in the leaf, we find leaves have a main vein which has lots of xylem in it. It has side veins branching off, and there are side veins branching off, side veins branching off. And you can see they're all of a different thickness, and that gives you an idea of how much xylem they actually have in them. Why is xylem important? Why is there so much xylem in the leaf? Another requirement for photosynthesis is water. So the leaf veins take the water to all the cells of the leaf so that they can photosynthesize. Next aspect is, why do leaves need stomata? We talked about the gas that is needed for photosynthesis, which is carbon dioxide. And the carbon dioxide has to go into the leaf, and it goes in through the stomata so that the cells inside the leaf can use the carbon dioxide for photosynthesis. Other substances also come out through the stomata. And that stomata basically allow gaseous exchange to occur in a leaf, and that is essential, or else photosynthesis won't be able to happen. We're now going to cut open a leaf and have a look and see where do we find the different tissues inside the leaf. If we cut a leaf open, we'll see something like this, because the leaf is long and quite flat. So, this is quite difficult to work out what's going on, but we always start with the easy bit. And what's the easy bit? It's got to have an epidermis. So there we have the epidermis. It also needs an epidermis on the lower part of the leaf. How do we work out what the next type of cells are? Clue: look for dark portions, because that is where the cells have got thick walls, and the thick-walled cells are usually xylem or sometimes sclerenchyma. So if we have a look, there are a whole lot of cells with thick walls, and that is where your main vein is, and that is where your xylem and phloem are. All these other cells here, those are the cells that are going to be doing the photosynthesis, the functioning of the leaf. Because the leaf is so large, when you look at it under a microscope, you can't fit the whole leaf under the microscope. We tend to look at it in two different sections. And this first section we're looking at is where you have cut the leaf through the main vein, where there's lots and lots of xylem and phloem. So let's have a look at this picture under the microscope. If we look at this picture, easy to identify. There is our upper and lower epidermis. Then, these big cells in the center, there is a patch of xylem and a patch of phloem. Then, have a look. Look at these cells here. What type of cells are they? What can we look for to help us identify what type of cells they are? We look for the thickened corners. So at the top of the vascular bundle and at the bottom of the vascular bundle, we have collenchyma, and that helps keep the leaf firm. You'll always find a bit of collenchyma around the vascular bundle. There's also a coating called a bundle sheath around the vascular bundle. So what about the rest of the leaf? We're going to be looking at the leaf blade. That's the flat part of the leaf where the photosynthesis occurs. And that's what we're going to be looking at in the next slide. We're not going to be looking at the main vein, we're going to be looking at the leaf blade or the flat part of the leaf. Sure, this looks quite confusing, but let's start logically. What can we recognize? We can recognize the upper and the lower epidermis. Oh my goodness, look here. There's a hole in the lower epidermis, and that's something we've looked at before. That is one of the stomata, a little pore through which the gas can get into the leaf. Then we find funny little patches here, and those are side veins or vascular bundles containing xylem with its thicker walls and phloem with its thinner walls. So what we can already identify is the epidermis and the vascular bundles. Vascular bundles bring the water, the epidermis does the protection, but the main function of the leaf is photosynthesis. So all the rest of these cells are involved in photosynthesis. If we look, we can see that the cells in the middle of the leaf, they're not all the same. The ones at the top are brick-shaped and tightly packed, and they are chock-a-block full of chloroplasts. That's why the top surface of the leaf is green. At the bottom here, the cells aren't regular shape. They're rounded, and there's lots of air spaces between them. But because these cells all have chloroplasts in them, they form what we call the mesophyll, the bits filling the leaf, and they're all different types of parenchyma cells, and because their function is photosynthesis, they are going to have lots and lots of chloroplasts inside them. Now, isn't this a much easier picture to be able to work out what is going on? But it's actually showing exactly the same as the previous picture. It's just because it's a drawn one, it's much, much easier to be able to identify exactly what is happening. Easy peasy. Upper and lower epidermis with a stoma so that gas can get into and out of the leaf. Also, we can notice on the upper and lower epidermis, we have a cuticle. Okay, so the epidermis itself is the cells, the cuticle is the waxy covering, and in the epidermis we have stomata. What else can we recognize? One of the small vascular bundles. At the top, it's got big cells with thick walls, and that is your xylem cells. And we know because they've got thick walls, and then phloem cells at the bottom. So that's your xylem and phloem, and surrounding it, that layer of cells surrounding it, that is called the bundle sheath. And these are the tiny veins that we saw when we looked at the leaf skeleton, showing all the different veins. Next thing we're getting to is the rest of the cells. Okay, and all of these cells inside the leaf we call the mesophyll, and they're all parenchyma cells. But because some of them are brick-shaped and others are rounded, we can divide them into two different regions. Okay, the top ones are called the palisade cells, or the palisade mesophyll. That's these ones here, and they are tightly packed like that. They're full of chloroplasts because their function is maximum photosynthesis, because that's where the sunlight comes in from here and falls on those leaves. The bottom cells, they don't get sunlight. They still have chloroplasts, but they don't get a lot of sunlight, so they're not as important in photosynthesis. The lower part of the leaf, where the cells are irregular shaped, is the spongy mesophyll. Important is they've got large air spaces, because even though they photosynthesize, their main function is gaseous exchange. And if we look at this diagram, this is the type of diagram you would probably see in some of your exams, where you would have to label the different parts and then explain what each of those parts actually does, what function it carries out. You might find that this diagram makes it a little bit easier for you to understand all the different parts of the leaf. It also shows quite nicely the leaf veins that travel throughout the leaf. What we have covered is we've looked at the structure of roots, stems, and leaves, and by now I think you guys need a break. Welcome back after your break. In this last segment, we're going to be looking at a little bit of what do leaves actually do? What is the function of leaves? And we'll be relating it to the structure of the leaf. Leaves carry out very important functions, and you can see them listed here: photosynthesis, transportation, gaseous exchange, and transpiration. We will be looking at those in detail in some of the next lessons, but I want to give you a brief introduction now. Important function, photosynthesis. We've been talking about that all the way through. Sunlight needed, water needed, carbon dioxide needed, food produced, and oxygen given off. How does the structure of a leaf lend itself to this? First of all, in your palisade mesophyll, that is where most of the photosynthesis occurs, and that is, just let me get a picture here, and that is because we need sunlight, and the sunlight hits the top of the leaf so that photosynthesis can occur. Water is also needed. Water comes out of the xylem and goes into the palisade mesophyll so that photosynthesis can occur. The spongy mesophyll, on the other hand, is mainly for gaseous exchange. So the gases will move in through the spaces and they will move up to the palisade mesophyll so that there is enough carbon dioxide for photosynthesis. Once the food has been produced by the palisade mesophyll, the food will diffuse out into the phloem, and the phloem will transport the food down to the roots. The last function of the leaf is the fact that it gives off water by transpiration. Some of the water coming up from the roots, some of it goes into the palisade cells so they can photosynthesize, but a lot of it actually passes into the cells and out through the stomata, and this process is called transpiration. In the next lesson, we will be having a look at transpiration and some of the aspects of water movement through the plant. So, what we have looked at so far in plant anatomy, we have looked at the structure of roots, stems, and leaves. And in doing so, we have also looked at the functions. What do these different parts of the plant actually do for the plant? In lessons going forward, we will be looking more specifically at leaves as the main organ of the plant, and we will be looking in detail at some of the functions of leaves, for example, transpiration, gaseous exchange, and water movement. I hope this lesson has helped you in your understanding of what a leaf looks like and why are these parts of the plant so important to us.